Hello everyone, this is Shami and I'm a junior majoring in biology.
Hello everyone, my name is Samantha Medina Alvarez and I'm currently in my junior year studying for my degree in biology.
Hello, my name is Ashley Osorio. I am a senior here at CSUB majoring in biology.
Hello, my name is Alyssa Montejo and I am a junior studying geology at CSUB.
Hello, my name is Natalie Nunez and I am a senior at CSUB majoring in biology.
We are part of McNish's Aquatic Ecology Research Lab participating in the SURE program. We are presenting our study here today on microplastic abundance pattern in Monterey Bay rivers and wastewater effluent. Microplastics are mainly known as an emerging contaminant of concern worldwide, mainly due to their ubiquitous presence in the environment and also their negative effects on wildlife and potential effects on human health.
Microplastics can enter the environment through different forms, including anthropogenic litter that break into smaller pieces, human waste, and from treated wastewater treatment plant effluent that enters rivers and oceans. Our research objective was to compare microplastic abundance from 3 freshwater river sites and from 5 wastewater treatment plant facilities that may be contributing to the abundance and transport of microplastics in the environment. Our prediction was that microplastics will be more abundant in wastewater treatment plant effluent than in rivers, with fibers being the most common morphology type. Our water samples were collected from the San Lorenzo, Salinas, and Pajaro river sites and were compared to 5 wastewater treatment plant effluent samples that were collected from the Monterey Bay region.
The samples from the river and the wastewater treatment plants were taken into the lab for further processing. They were fractionated by size class of 1,000 to 50 and 20 micrometers, and they were later digested with a hydrogen peroxide of 30% concentration at 45 degrees Celsius, as you can see in photos A through C. The samples were also then vacuum filtered into 1.2 micrometers filters, and we used rose Bengal dye to identify microplastics from organic materials, as you can see in photos D through F. The dye filters were examined with a microscope to count microplastics and categorized particles by shape and morphology, as you can see in photos G through H.
We did a descriptive statistics table to compare microplastic abundance in wastewater treatment plants and rivers and observed that wastewater treated plants had greater microplastic abundance of 1.5 compared to river habitats. The wastewater treatment plants had a maximum value of 55 particles, while in the river samples, the maximum value was 16 particles.
The relative abundance of microplastic morphology was calculated comparing wastewater treatment plants and river habitats. The 4 morphological categories that were present in our samples were bundles, fragments, natural fibers, and plastic fibers. Natural and plastic fibers were the most common microplastic found in both habitats. Microplastic morphology patterns, however, were not statistically significant between habitats. The relative abundance of plastic and natural fibers within wastewater treatment plants and river habitats was calculated. Color patterns of the natural and plastic fibers were not statistically significant between wastewater treatment plants and river habitats. However, blue and clear colors were the most common colors across both habitats and fiber types.
As shown in figure 3 and 4, the microplastic abundance, morphology type, and color categories were not unique across the wastewater treated plant effluent and river habitats, resulting in our prediction being partially not supported. However, microplastic particles were more abundant in wastewater treated effluent samples compared to rivers, which partially supported our prediction.
Wastewater treatment plants account for the elimination of 77 to 99% of microplastics. However, due to the large quantities of microplastics that enter wastewater treatment plants, a significant number of them are released into aquatic habitats that can then interact with biota and potentially cause toxicological hazards to the environment.
This study can be further developed to include multiple sampling time points across ecological seasons to identify microplastic patterns across temporal scales that could potentially be linked with changes to seasonal availability of water.
We would like to give special thanks to the Chevron for the financial support allocated towards this research to happen. We would like also to thank the Department of Biology of California State University, Bakersfield, for giving us the chance to conduct our research in their facilities. We would also like to thank our collaborators for collecting field samples and also appreciate McNish Lab members who helped in data collection. Also, we would like to thank the additional funding that was provided by the U.S. Department of Education Title VB grant. Thank you so much for attention.